Okay, so today we'll study the chromosomal aberration, that is numerical aberration, and then if we finish this one, then we will go for the DNA structure. Okay, so this numerical aberration means the change in chromosome number, either one or two or the whole set. When we'll be talking about the whole set, that means it is multiple of n. So n means in case of Drosophila, n means 4. In case of human, n means 23. So there are mainly two kinds of things. One is called aneuploidy, another is called euploidy. In your book, you will find chromosomal numbers, that is just polyploidy and aneuploidy. But generally, we are grouping this one as euploidy, the polyploidy part. Okay, aneuploidy means just a change in one number. So these are the things. Monosomy means one chromosome is missing. And try to think about this one. One chromosome is missing means we have 23 pairs of chromosomes. That means 46 chromosomes. If one chromosome is missing, that means it will be 45 chromosomes. Okay. One chromosome is missing means any of the chromosome from any of the pair is missing. Trisomy, that is in case of haploid only. So when there are haploid number of chromosome and one chromosome is getting 2 in number, then it is called disomy. Trisomy, we have 46 chromosomes. Suppose chromosome number 21 in 3 numbers. So generally we will have chromosome number 21 in 2. If there are 3, then in case that is called trisomy, Down syndrome. Trisomy of 6 chromosomes, XXY, that is the trisomy. So total number of chromosomes will be 47 in case of 46. Tetrasomy, 1 chromosome in 4. So we will have 2, that means 1 pair, but there are 4. So that is called tetrasomy. So only the change in number of 1 chromosome. That is called aneuploidy. But what is the difference with this euploidy or polyploidy? The whole set of chromosomes. Whole set of chromosome means we have n. Suppose n for us is 23. We are diploid, that means 2n is equal to 46. If we have 3n, 23 into 3, the whole set will be triplicated. That is called polyploidy. So triploidy means 3n, that means in case of us, that is 23 into 3. In case of Drosophila, it is 4 into 3. In case of some other species, that is their number into 3. So the whole set of chromosome, we have 23 chromosomes in 3 times. Clear difference between these two? So there are some other things, triploidy, tetraploidy, that is 4n, pentaploidy, 5n. Generally, these are very much popular in case of plants and some fish and some other lower organisms. But generally, in higher organisms, we don't see that much of polyploidy, generally. Another thing is that monoploidy. So it was my question. What is the difference between haploid and monoploid? Haploid means n, monoploid means n. What is the difference between these two? Monoploidy means half set of chromosomes, but it is abnormal. In case of haploidy, generally that is the meiosis, and as a result of meiosis, we are getting half set of chromosomes. That is haploidy. It is normal. But in case of monoploidy, it is abnormal. Now we will talk about those things, how these are occurring in nature. And that is the main thing is called non-disjunction. And this non-disjunction means generally, we know during meiosis we have prophase, metaphase, anaphase, and telophase. During metaphase, the spindle fibers are attached to the chromosomes and chromosomes are aligned to the equator pole. Okay. In case of meiosis 1 and meiosis 2, the difference is that in case of meiosis 1, the homologous chromosomes are attached there. In case of meiosis 2, chromatids are attached there. These are the differences between these two. But after that, the next step is the that is movement of chromosomes to different poles. That is called the anaphase movement during anaphase. But if the spindle fibers are somewhere, it is destroyed or something like that, then the chromosomes can't move to the pole. That is the main reason for non-disjunction. There are several other things that we know into the cell cycle that there are some checkpoints. Okay, during anaphase, if the chromosomes are not attached to the spindle, then the cell will not divide. That is the cell cycle regulation or cell cycle control. So in such case, in one cell, we'll have double number of chromosomes. And that is the non-disjunction. So in such case, what is happening here? Suppose we have two chromosomes. Yes, sir. During anaphase, what will happen? I am not doing the tetrad formation, just the two chromosomes. Okay. It will be half after meiotic division 1. So in normal case, we will have after first meiotic division, we will have like this. This is the MO. Okay. After second meiotic division, we'll have this is just doubling of the chromosomes. That is the 4 sets of chromosome 1 each. This is after second meiotic division, right? Now, in such case, if these two chromosomes are not separated during first meiotic division, one spindle fibers in one pole, it is destroyed, or particularly for one chromosome that is destroyed. What will happen? In such case, one cell will contain two chromosomes, other cell will not contain that chromosome. Right? Try to understand this point. If it is a single chromosome, that is possible. If it is a whole set, that is also possible. In one set, all the chromosomes are there. In another set, that chromosome is not there. So when there will be another division in meiosis 2, so we will have this kind of gametes, the 4 gametes, because that is just the division of those gametes into 2 in the second meiotic division, just like mitosis. So we will have this kind of gametes. This is one possibility. Another possibility is that there is no problem in meiosis 1. There is no non-disjunction in meiosis 1. But non-disjunction is there in meiosis 2. What will happen? If the non-disjunction is here, then we'll have this kind of configuration. Is it possible? Here, the chromatids are not separated. They became one chromosome. So in one cell, all the chromosomes are there. In another cell, there is no that either that chromosome or not the whole set. So these are two possibilities for non-disjunction. So non-disjunction means that during anaphase, if the spindle fibers are destroyed, generally this is one more common thing. Then in one cell, the two sets or two numbers of chromosomes will be there. They are not separated in two cells. In one set, they will contain all the chromosomes. In another set, there will be no chromosomes. So this is the formation of gamete. Now think about Klinefelter syndrome. So Klinefelter syndrome is X and Y. Okay, this is the final one. So that means two gametes made together to form these things. If the female gamete, non-disjunction is here in meiosis 1. So what will happen? The 2X will go to one cell, and in another cell there will be no X. All other 22 chromosomes are there, but there is no X. So it is 0, or this is called null gamete. If that chromosome is missing, this is called null gamete. So in such case, we'll have this 0 here, and there is no non-disjunction here. So in such case, we'll have right or XX. What do you think? XX or single X or double X here? Double. Because first meiotic division is the halving of the chromosome, second meiotic division is just like mitosis. Okay. So we will have this gamete, this abnormal gamete containing two X chromosomes. Okay. From male, we have XY condition. So from XY, we can have one X and one Y halving of the gametes, that is the chromosomes. Then from here, we'll have two X gametes and two Y gametes. Now, if these two fuse together, what we'll have? We'll have this XXY condition. So from male, we need Y gamete. From female, we need this abnormal gamete, and we can have this kind of Klinefelter syndrome. Okay. Now, suppose this null gamete, it contains no X chromosome at all. If this fuse with this one, what will have? We'll have one X, and 1 0. That is the X0 condition. This is Turner syndrome. Okay. Suppose rather than XX, this chromosome number 21. Just think about chromosome number 21. So 21 is there. Here is 21. Here is 21. From male, we are having 121. And if they joins together, there will be 321 chromosomes. That is Down syndrome. Clear? Now, non-disjunction can be of meiosis 1, can be of meiosis 2. Non-disjunction can be in male, can be in female. If the female condition is okay, female is producing the normal gamete, what will happen for the male? So these are normal gametes. But here is non-disjunction in meiosis 1. So we'll have one this gamete and one gamete is null. This gamete will be XY. If this XY will meet with this X, then it will be Klinefelter syndrome, XXY condition. If it is super Klinefelter, then that means 2X, 2Y. Then there is a chance of both of these parents can have non-disjunctions in their gametes. Or a Klinefelter syndrome patient can have the gamete. Generally, they are not fertile. If they are fertile, if they can produce gametes, then there is a chance that this is a Klinefelter. If this one fuses with normal X gamete, then we will have super Klinefelter. If this fuse with normal Y gamete, then we will have this condition. Or in case both the parents have non-disjunction. So one from here we will find one XY gamete, from here we will find two X gametes, then if they fuse together, but those chances are very rare. Because non-disjunction is not a common phenomenon. It is a rare thing. And those chances are very rare. I will suggest to practice this one. Just form the Klinefelter syndrome using non-disjunction in first meiotic division of female, second meiotic division of female, non-disjunction of first meiotic division in male, second meiotic division in female. 4 combinations, and try to do that one, practice it with the formation of Klinefelter or Turner. Okay. then it will be clear. Then this is a very most important thing, particularly for the genetic diseases. So for the aneuploidy, the same thing. If one chromosome number is different, then it is called the aneuploid. Nullisomy means both chromosomes are missing. That is the null gamete. What we are forming here, that is the null gamete. Both chromosomes are missing. That is called nullisomy. Monosomy means loss of a single chromosome. In such case, only one chromosome is there. That is called monosomy. That is 2n minus 1, 45, Turner syndrome. Trisomy, gain of one chromosome, Down syndrome. Chromosome number 21 in 3 times. Tetrasomy, generally more, and that is in case of plants. In human XY, that is super Klinefelter is also there, tetrasomy. So, couple of sex chromosome aneuploids in human XXY condition, XYY condition, because in such case, if the Y gamete is there is a non-disjunction here in the Y gamete, we can form here 2 Y chromosomes. Suppose this is X and this is Y, and here is non-disjunction. Then we'll find YY condition. If that YY gamete fuses with X1, we'll find XYY condition. XXX, one from parent, one parent, there is non-disjunction having 2 X gametes, and one parent have only single X gamete, then we can find XXX condition. And X0 with fuse with the null gamete. Normal gamete X with the null gamete, that is the X0 condition. There are a couple of examples here. So what is the answer of this? Just a very simple. When is equal to 36? What will be in the trisomy?
37.
37 or 18 into 3. What do you think? 18 into 3 or 37? This is trisomic. Okay, that means aneuploid. Only one chromosome will change. So it should be 37. If it is triploidy, then it will be 18 into 3. Okay, this is the difference between these two. And we have some kind of that is phenotype changes, particularly for the aneuploidies, and these are a couple of examples. Generally, mostly we can find this in plants. And this cone shape, there are different types of cone shape, changes in datura, and that is for different kinds of trisomics condition. Only one chromosome is double there, and based on that one, we have this kind of conditions here. Okay. Trisomy of 21, that is the Down syndrome. And this is the karyotype and showing that number 21 chromosome is in 3, and each and every other chromosome in set, 2 of that. Generally, Down syndrome is the 75% random non-disjunction in egg formation. Generally, particularly for the females, and it is the trisomy of chromosome 21. Generally, in case of every genetic disorder, so every genetic syndrome, mainly people are not fertile, generally sterile. Because why? The question is why? They are generally sterile, not fertile. This is a very common. Again, think about meiosis. During meiosis, there the chromosomes, homologous chromosomes, should pair with each other. That is the first part of meiosis. Homologous chromosomes should pair with each other. But if one chromosome is in excess in number or one chromosome is missing, they cannot pair. There is no homologous pairing. In such case, cell cycle regulation will stop the meiosis, the cell division. Because we know the cell cycle checkpoints are there. If the chromosomes not are properly attached, if the chromosomes are not properly present, if the spindle fibers are not properly attached, cell cycle regulation will stop the division. So in such case, there will be no division, there will be no meiosis, and they cannot form any kind of gametes. That's why mostly they are sterile, mostly. But sometimes if they pass the checkpoint in some other way, because totally this is totally biochemical process, and it is not perfectly controlled by everything, they can pass sometimes, then they could be fertile. But most of the cases they are non-fertile or sterile. So one is that, and the trisomy of 21, and you could remember another thing that we have, we learned about Robertsonian translocation. What is Robertsonian translocation? That is the part of this chromosome, that's chromosome number 14, one of these chromosome is attached to this one, they will form the trisomy of 21. Most of the 21 part is present there, only very small part of 14 is present there. In such case, most of the genes of 21 is there. In such case, they will form the another extra copy of chromosome 21, and that could cause the Down syndrome. So Down syndrome can be caused by two ways, either the normal aneuploidy, that is the chromosome number 21 in 3, or Robertsonian translocation. And these are the different ways, that is 1421 combination, that is the Robertsonian translocation. In such case, if those parents have this kind of things, how they can give this chromosome to their offspring. So this is the translocation carrier. They can give birth. They can give birth, but might have a chance of Down syndrome. Okay, but generally, if there are so many options are available here, but generally, if the chromosomes are not properly attached or the gametes are not properly formed, they are generally aborted the first time. Okay, they cannot form the full embryo anyway. So in the population, we cannot find all those things. Trisomy of 13, Patau syndrome, and trisomy of 8, there is no such things, but there is another thing is there, and it is almost 1 in 25,000 to 1 in 50,000, very rare. But in case of those two, those are not very rare as compared to this one. But Klinefelter is, in such case, Klinefelter is a little bit more common than this. And generally, these type of patients, they don't survive more than 20 years, because I know the Klinefelter is 20 to 25 years, like average lifespan. But for other cases, it is not too much. Towns and Turners also have that kind of things right now, because we have some kind of treatment available to just for their survival, but we cannot change anything. There is no way. That's why that is the question right now, that whether if we can have some kind of gene therapy, that we can change the chromosome structures. Okay. Okay. But this aneuploidy, it has, we observed that during the age of mother, if the mother's age is higher at time of birth, there is a more chance of aneuploidy than in normal cases. Okay, it has been seen that. That's why we have some kind of optimal age for the child birth also. It is not very early. Generally, it says that females between 18 to 35, that is the best way. After 35, there are a lot of complications, not only the aneuploidy, but there is a very good chance of cancer. And we have found that thing. Okay. Now, this aneuploidy can cause some different things, particularly in case of Drosophila. If some of the cells have that kind of aneuploidy, so they can form this kind of mosaicism. This is XX X0 condition. XX X0 condition, that means in X0, where the white eye is the mutant type and red eye is the normal type. In X0 condition, the white type is prominent. In XX condition, the red type is prominent. Some cells contain XX, some cells contain XY. In such case, in one offspring, you can have two characters. One side the eye is red, another side eye is white. This is called genetic mosaicism. And generally, you have seen that in cat also, that is the orange fur color and red fur, that is black fur color. That kind of thing, that kind of mosaicism is present in case of lower graded animals, not the higher animals. Mainly in Drosophila and some kind of sea urchins and those kind of things. Okay, this is called genetic mosaicism. So in one side, the male expression is there. In another side, the female expression is there. In one side, the red eye color. In another side, the white eye color. Now we need to think about the whole set of chromosome duplication. The whole set. There are two things: autopolyploidy and allopolyploidy. Autopolyploidy means that chromosomes are from single species. Only one species, the chromosomes are there, and these are very common in plants. So, using this that is phenomena, we have different kind of wheat varieties. Okay, allopolyploidy means from two or more different species. So these are the things that during mitosis they can occur through mitosis or meiosis in plants. Through mitosis, they can change the separation of sister chromatids. Up to this point, they are okay, but there is no solution. So in one chromosome, they have autotetraploid cell. Division is there. Chromatids divides into two. One chromatid divides into two. That is the replication process. DNA replication is there. Everything is two in cells, but there is no cell division. Cell in one cell is there. No cytogenesis. In such case, one cell contains four sets of chromosomes. That is during mitosis. In case of meiosis, there is a chance of non-disjunction. Either in meiosis 1 or in meiosis 2, they can have this tetraploid condition. And these are different patterns that how these chromosomes can separate in which condition, and they can form different kind of tetraploid or triploid conditions. Okay, just go through this one. This is simply like non-disjunction part. These are three things here, that how the chromosomes are arranged during meiosis, that is the spindle formation. Based on that one, in which part they are separating, that is creating their tetraploid, polyploid conditions. Okay. And allopolyploidy means there are two different species. And here is one thing, that there is one species here having three chromosomes, another species having three chromosomes. When they are forming gametes, each in one, so that is the haploid condition. So they have three chromosomes in one dose, they have three chromosomes in one dose. Both species. Okay. When they combine together, they have all six chromosomes, but in one dose. In such case, there will be no chance of formation of gamete. In such case, if the DNA replication is there, but there is no such separation, that will double the chromosome number. DNA replication means doubling the DNA amount, doubling the amount of DNA or chromatids. In such case, there will be chromosomal doubling. And it happens in plants sometimes. In such case, if the chromosomal doubling is there, in such case, that the replication was there, but there was no such division and the chromosomes duplicated. Now they can separate into gametes in this condition from two different species. Then there is no division and as a result, there is chromosomal doubling. This is called chromosomal doubling. When the chromosomal doubling is there, they can be fertile. And using this condition, this chromosomal doubling, we actually created a lot of varieties of plants, particularly wheat, and this is for something sunflower. We have created this kind of things. And when the chromosomal doubling is there, the number of the and size of the cell is bigger. And using that thing, we have created a lot of fruits and other things with the bigger size. It is often associated with the increase in cell size because if the chromosome number is more bigger, then the volume of the nucleus will be bigger, then the cell will be bigger. Using this attribute, we can have plants with larger leaves, flower, fruits, and seeds. But this is very uncommon for human, not only human, in the higher animals, mostly common in plants. And that one, if you go to this picture again, this individual with after chromosomal doubling, they are fertile, but they have different type of characters than that one. Okay, so they may arise into a new species, and that can be possible. And new formation of new species means that is evolution. Do you know that one? What is evolution? What does it mean, evolution?
Pass on genes that are more evolved or can survive, and the other genes kind of die off, so the organism advances.
Evolution means formation of new species. That is the first thing. So, what is species? I told you before. What is species? We were gorilla, right now we are humans. We were apes before, right now we are humans. So we are new species. What is new species? A group of animals or plants who are reproductively isolated with other. They are called the species. And when the species is formed, that is called speciation, and the formation of new species means evolution. So in such case, if that is a new type of species that can be fertile only between themselves, not with their parents, then it is a new species and that can arise in, that can result in evolution. So this is one example. I just put it here for our wheat. And that is simply the same thing with two different species of wheat. We made together, then there is chromosomal doubling, and after the formation of chromosomal doubling, we have a new species with a greater variety of wheat. And there are a couple of other things: potato, banana, peanut, sweet potato, tobacco, cotton, wheat, oats, sugarcane, strawberry. We have this kind of allopolyploid. And that is in for beneficial for our agriculture purposes. What about this? Answer. So allotriploid means one in full set, another in half set. So 14 plus 8, that is one, or 16 plus 7, another one. So this is all about chromosomal variation. So there are two things: structural and numerical. For structural things are very important because in case of point mutation, we need When we will go for DNA replication, we need to think about all those things. So structurals, there are four things: deletion, replication, uh, translocation, and inversion. And for numerical aberration, we have aneuploidy, euploidy. In euploidy, we have monoploidy and polyploidy. So monoploidy means if there is a chance that chromosomes, there is no formation of spindle fibers, and there is no non-disjunction there. This is the difference between haploidy and monoploidy. Clear? So this is more or less all about your transmission genetics or hereditary part. Now we will go to the molecular part. Okay. That means the structure of DNA, DNA replication, transcription, translation, and all those things. So at first we need to think about that during this period, this transmission genetics and when all the process and everything was done during that time, we really didn't know about the structure of DNA or all other things. We know that Mendel's factors were there in the cell, and where are those factors? Then we have found that okay, it is in the chromosome, and from chromosome we know that there is something in DNA, but still we didn't know that what is the structure of DNA and what is called gene or all other things. So we named that as gene. Now, what is genetic material? Genetic material has some kind of properties. That is, it must contain complex information. Complex information means the information of different characters. Different characters. And the larger information, definitely, because we have almost like our DNA is 6 billion base pairs long, having 21,000 genes. Bacterial DNA contains about 71,000 genes. Okay, so large information should be there. Must replicate faithfully. So each and every division, either it is mitosis or meiosis, it will replicate the DNA. The first part is that the replication of DNA. One chromosome needs to form another chromosome with the same homologous condition. Okay, genetic material must encode the phenotype. So not only they have these genes there, they must express. What is the expression? They need to produce mRNA, they need to produce protein. Proteins are the expression of gene. That is the final result. And that genetic material must have the capacity to vary. Capacity to vary means we are from our parents, our father and mother. So we must have a combination of those two characters, right? But we are not. We are not. We have some kind of similarity with our dads, with our moms, but we are not identical to them or not in middle of them. Okay? We have certain kind of variations. Why? Because of crossing over. During crossing over, the genetic material is exchanged between chromosomes, and that's why the variation occurs. So they must have some kind of variation. If there is no such variation, if there is no such mutation, then the species will not survive. They will die. That is the normal condition. And this is called inbreeding and speciation. When we'll talk about this population part, we'll talk about that one. So these are a little bit history of DNA or history of the that is discovery of DNA. Just go through that one. I will not talk too much about this, but we need to talk about a couple of things. The first thing is that Chargaff's rule. During this time, during 1930s, 1940s, people were thinking about the DNA, what is DNA, how, what is the structure of the DNA, what are the materials there. They have found that there are four nucleotides. One is adenine, thymine, guanine, and cytosine. The first time Chargaff found that each and every species, this ratio is different. Okay, and the fine thing is that how many A is there, adenine is there, how many that much of thymine is there, the amount. They didn't find out the actual things. He just found out the total amount of adenine, total amount of thymine, And each and every species, he have found that adenine and thymine is more or less similar, cytosine and guanine is more or less similar. Okay, at that time we didn't know about the structure of DNA. And each and every species, that number little bit varies. That is different. The number amount of AT and amount of GC is different. But right now we have the explanation of this one. What is the explanation? Why the A amount of A is equal to amount of T and why the amount of C is equal to amount of G? What is the explanation of this?
They pair. A pairs with
They
T.
pairs together. A cannot pair with C or G and T cannot pair with C or G. So the first thing is that we have to find out the DNA is the genetic material. That was the first question, whether the DNA is the genetic material or some other protein is the genetic material or carbohydrates is the genetic material. We didn't know that one. Based on that question, we have a couple of experiments here. The first one is for Griffith. What is that? He was working with Streptococcus. And Streptococcus is a bacteria causing pneumonia. Okay. And if the bacteria can cause the disease, the pneumonia, it is called virulent type. And he has found couple of two different types. One type is not causing the disease. That is called the non-virulent type. So this one is the virulent type and this one is the non-virulent type. So if I inject the virulent type bacteria into mice, the mice will have pneumonia. If I will inject the non-virulent type of bacteria into mice, they will not have any kind of pneumonia. Okay. Now, those virulent types, they have a very good surface. Surface is smooth, those bacteria. They have very smooth surface. But non-virulent type, they have a rough surface. Rough surface means some kind of carbohydrate or protein part is missing on the surface. There are two different things he has noticed. That is the first thing. First thing is that virulent and non-virulent type. Virulent type has the smooth surface, non-virulent type has rough surface. Now, these two types, this is called type 3 and this is called type 2. To change from type 2 to type 3, it requires a lot of mutation to change that. Okay, so these are two different kind of bacteria. Okay, now the question is that when he put this bacteria, that is the virulent type of bacteria, into mice, the mouse dies because there is pneumonia. He put the non-virulent type in the mouse lives. There is no pneumonia at all. Now he did some experiment with this bacteria and he put some kind of high heat onto the bacteria, and as a result of the heat, they can kill the virulent type bacteria. Okay, they can break the surface of those things. So this is called heat killed bacteria. Virulent type, heat killed, that means they will not cause any kind of pneumonia in mice. Virulent type generally cause pneumonia, non-virulent type not causing pneumonia. Heat killed virulent type will not cause any kind of pneumonia. Okay. So the heat killed bacteria, when he injected those into mice, they will not cause any kind of pneumonia, so they live. Now he put those two types together. Non-virulent type, they will not cause any pneumonia. Heat killed bacteria, they will not cause any pneumonia. So these two non-virulent type he mixed together and put into mice, and the mouse dies. So what is the cause behind it? Because heat killed cannot cause pneumonia and virulent type, non-virulent type cannot cause pneumonia. But when they are together, they caused pneumonia. So what is the explanation behind it? The The explanation is that non-virulent type generally they have something, and if they transfer that material, or the virulent type they have something, they transfer that material to other materials, so they can become the virulent types. Okay, these two non-virulent types, they can transfer some materials to each other, they become virulent types. These material, the exchange of this material is called the transforming principle or transforming material. So that is the first time they have noticed that there is something is there which can transform the non-virulent type bacteria into virulent type by in this way. So his experiments what then later that transforming principle is there, transforming material they have identified. Now the question is what is that transforming material? Is it a protein? It is a DNA? It is RNA? Or what is this? Then these three scientists, Avery, MacLeod, and McCarthy, they did this experiment. Just like starting with the that is the Griffiths experiment. First, heat kill the bacteria, the same bacteria, Staphylococcus. Then filter this bacteria, separate out all the components. Okay. The first thing one here, take out the type 2 bacteria, just destroy the RNA. There is no RNA there. And another one, use the protease, destroy the protein. So RNA and DNA there. Another one, destroy the DNA. There is only RNA and protein. So first one, treat with RNAs. RNAs means that will destroy the RNA. So we have the DNA and protein here. This one with the protease treatment, that means destroy the protein. So we have the DNA and RNA there. And here, destroy the DNA, only RNA and protein is there. Okay. Now, just like Griffith's experiment, this part add to heat kill type. That is type 2 bacteria. So we took the heat killed one and we used that type 2 bacteria. So non-virulent type, add that one and check the growth of that. In the first one, we got the virulent type bacteria. What was there? We destroyed the RNA, so DNA was there and protein was there. So from this part, we can conclude that either DNA or protein can change this material, or that is the transforming material between these two. Clear? Only from this one. The second one, we destroy the protein. We have DNA and RNA. We already destroyed the DNA, RNA here. So RNA is not causing this transformation. That is clear from here. Here we have the DNA and protein. So that DNA and RNA. So if RNA is not causing that one, that means DNA is causing that one. And here we destroy the DNA and it is not causing any kind of transformation. So these are the three steps which is saying that DNA is the genetic material. Clear this part? Try to understand in this way. The first one, protein is not there, RNA is not there, so either DNA or protein. Second one, protein is not there, that means it is DNA. Because RNA is not causing anything. We already did it from first experiment. The third one, DNA is not there and it is confirming that it is DNA. Okay. So after this experiment, we knew that the transforming material is DNA. And DNA is the genetic material. Another experiment was that in T2 bacteriophages. What is bacteriophage? What is that? Do you know phage virus?
It's a virus that attaches to the bacterial cell and the bacteria becomes its host.
Yeah. Generally, it is a virus, and the structure of the virus is something like this. It has a protein capsule. This part is protein capsule, and all other some kind of limbs are there. These are made up of protein totally. In this whole protein part, in the top of that one, there is a cell that is a capsule. It contains only DNA or RNA. Okay, the nucleic acid. So there are two things here, either nucleic acid or protein. Only two things. What is the function of this one? When they are attacking the E. coli, they are generally attacking the E. coli bacteria, Escherichia coli bacteria. And this virus, when they are attacking those bacteria, they transform their genetic material to this and break down the genetic material of the bacteria. They have the circular DNA, they can break down that DNA and replicate using that material, they can replicate by themselves. That genetic material is replicating by themselves. And into that bacterial cell, they can form their own structures, like their capsules and their stalks and other things. So they can replicate by themselves, they can form new viruses from there using that bacterial DNA. This is called their infection. Generally, when we have the viral infection, it is something like that. Okay. Now the question is that based on this principle, they did one experiment and they are Hershey chase. This experiment is very, very important. Hershey chase experiment. Okay. Now, what is there in DNA? In DNA, we have nucleotides and phosphate. What is there in protein?
Amino acids.
Uh, did we go for the protein structure? Do you know the protein structure? Primary structure, secondary structure, tertiary structure, quaternary structure of protein? I think we didn't discuss here because we were in genetics from the before. But in your previous class, do you have any experience about that one? The structure of protein? No? Okay, so protein has four structures. Protein is made up of amino acids. Okay, so those two amino acids together, they are binding with each other. These amino acids are bound by one bond that is called peptide bond. So it is something like two amino acids binding together by peptide bond. This is called the peptide bond. Okay. When they are in this kind of linear structure, this is called a primary structure of protein. Only the peptide bond is. In the secondary structure, they can form some kind of hydrogen bonds between them. H bonds. Okay. In such way, they can form a seat-like structure. This seat-like structure is called the secondary structure of protein. Okay. After this one, they can form three-dimensional structure, and three-dimensional structure is formed between the sulfide bond between those two proteins. Either with the primary structure or secondary structure, they are together and they form the sulfide bonds, and they can form the tertiary structure. Now, when these two tertiary structure, primary structure or secondary structure, two units are there, they are making together to form a protein that is called quaternary structure. Like hemoglobin. In hemoglobin, we have the heme group, that is the iron, and the globin part, alpha globin and beta globin. These two globin subunits are together, heme part is together as a whole, that is a protein, that is called the quaternary structure of the protein. Okay. Why do we need to know that? Because in case of tertiary structure, there is one bond formation, that is the sulfur bond formation, sulfide bond. In protein, sulfur is present, but in DNA, there is no sulfur. In DNA, there is phosphorus. In protein, there is no phosphorus. Okay, these are two main important parts. Now, what he did that, he took out all those materials, DNA materials from there, from the phage virus and from the infected E. coli. Then he labeled all those proteins and the DNA. Okay. So from phage virus, we can get two parts. One is protein, that is their capsule, their body structure, everything is made up of protein. And another part is the DNA, and that is the nucleic acid containing phosphorus. Protein part sulfur. If you use the sulfur, radio level sulfur, and that is what is 35S. 35S sulfur, that is radioactive sulfur. If you incorporate that sulfur in a culture medium and you can grow the bacteriophage there, they will incorporate this sulfur into their proteins. So their protein will be radiolabeled. Clear this part? So in one case, they radiolabeled this bacteria or the viruses with the 32S sulfur. So all the proteins are containing 32S sulfur. In another part, he used 32P, that is radioactive phosphorus. This phosphorus will go to DNA, okay, because DNA contains phosphorus, protein contains sulfur. So in DNA of those bacteria, okay. Now they were infected, all those phages, they were E. coli were infected with all those things. Then they transferred their genetic materials to the E. coli bacteria. Okay, that's fine. Now the question is that if they transferred the protein, then the E. coli bacteria will contain that radioactive protein. Okay. If they transferred the DNA there, then the E. coli bacteria will contain that radioactive DNA, because there is no source for the E. coli. We just added the phase with these two type of things. Okay. Now we have to find out that this infected material in E. coli, what we have. There are some procedures that the infected E. coli, you have to separate that one, you have to homogenize that in, and you will get all these proteins here at the bottom of the tube, and then find out what is there. Generally, it is not found that radiolabeled protein is there. Okay, so that means radiolabeled protein is not infecting the E. coli. So they are transmitting some particle, but that is not protein. If I will not find this one. Clear this part? In this case, when the DNA was radiolabeled, in that E. coli bacteria, we got the radiolabeled DNA, that is the phosphorus. That is indicating that the DNA is transferred into the bacteria, but protein was not transferred. So what is the transforming material? That is the DNA. Clear? These three experiments, these are very, very important experiments, particularly for the base of DNA on nucleic acids. Now tell me this one. This will clear your concept. What do you think?
Say A.
A.
Protease is an enzyme.
Okay. Any other? Try to think a little bit. Is AAC, if it is there, that means that is an enzyme. The first thing. That is enzyme. So found that heat-killed bacteria treated with RNAs and DNAs transformed bacteria, but samples treated with protease did not. Sample treated with DNAs destroy the DNA, but it is showing the result. Treated with RNAs, that means destroy the RNA. DNA is there, protein is there, that means their result is okay. But protease, that means destroying the protein. Actually, that will give us the result here. In this case, this is hypothetical. So that means if protease is there, when we are treating with protease, that means we are destroying protein, then we are not getting any result. What does it mean? This will be your questions. So at first, DNAs treated with DNAs. That means DNA is not here, only RNA and protein is there. Treated with RNAs. That means DNA is there, RNA is not there, and protein is there. From these two, what we are find? Which one is common? If DNA is not there, then there is no transformation. This is not with the actual experiment. Just read it very carefully. If DNA is not there, but there is a positive result, that means it could be either RNA or protein. Okay. Now RNA is not there. Already we got the DNA is not there. Now it should be the protein. The confirmation test is when we are treating with protease. That means DNA, RNA, and protein is not there. Then we are getting the no result. That means it is confirming that it is protein. This is the logical test. Okay. And this is if, not the actual. In actual thing, the DNA is there. Now, DNA as the source of genetic information. All the genetic information are in DNA or RNA. And Watson and Crick at first discovered the structure of DNA. DNA was discovered before the structure of DNA, how the DNA looks like, and what are the things there in the DNA. Watson and Crick at first discovered the structure of DNA in 19. Okay. So what is X-ray diffraction image? That if you have some material and if you pass the X-ray through it, you will have all the components, the picture of the components, how they are attached. And from this image, actually, what the Franklin actually got, she has found that there are some similar structure in both areas. There are two strands like things, and they have more or less similar structures. This pattern. Okay, that is the first thing she discovered. This person, but she didn't get that recognition that we have these kind of things. And actually, she had the idea that DNA is double strand. That is the first. From her result was taken by Watson and Crick, and they started thinking about this pattern. What is the structure of DNA? And they have found that according to her X-ray diffraction image, the DNA is double stranded. Okay. Now, that is one thing. The DNA has genetic material. In some kind of organisms, there is only RNA. There is no DNA there. They are getting the genetic information as well. And that is one thing is the tobacco mosaic virus. And in this experiment, we have RNA and protein. And when we take out the protein in the genetic material, not the proteins. Now, in DNA, there are two main things. The base or the structure of DNA is like that. They have a nucleotide base, a phosphate group, and a sugar. There is a sugar, and this sugar is the pentose sugar. With this pentose sugar, one base is attached, and here is one phosphate group. This is the basic structure of DNA and RNA. Based on the difference between these sugars, either this is deoxyribose sugar, then it is DNA. If it is ribose sugar, then it is RNA. Okay. And this is the difference between chemical structure difference between ribose and deoxyribose. Generally, the base one, the carbon atom attached to the base is the number one. And here is the 3' position, the third position, where is the phosphate group. And this phosphate group is attached to the 5 position of the carbon sugar. In 5 position, there is OH group here. This OH group is attached to the phosphate group in the next nucleotide base. This is the pattern, that's why it is called 3' to 5' position. Okay. Based on these structures, there are two types of nucleotides. One is called purine, one is called pyrimidine. Purines are that having two, that is ring structures. Two ring structures, there are two only nucleotide. One is called adenine, another is called guanine. Okay. Pyrimidine, there are three types. One is cytosine, thymine, and uracil. Cytosine and thymine is present in DNA. Thymine is only present in DNA, and uracil is only present in RNA. This is the difference between DNA and RNA. So there are two differences. One is that their sugar is different, deoxyribose sugar and ribose sugar. In case of RNA, uracil is present instead of thymine. In case of DNA, thymine is present instead of uracil. Now, they contains the phosphate group here also. When they are containing one phosphate, that is called the monophosphate condition. Generally, when they are free in cytosol, they are having 3 phosphate groups. That's why they are called triphosphate condition. So adenine with one phosphate is called adenosine monophosphate. Adenine with 2 phosphates, they are called adenosine diphosphate. Adenine with 3 phosphates, they are called adenosine triphosphate. Adenosine triphosphate, guanosine triphosphate, uracil triphosphate, they are generally free in nature, free in cytosol. And that is required for the replication of DNA. One thing is here, when Watson and Crick was trying to fit the model of DNA, they have this idea there is purine group and there is pyrimidine. The question is that if those two purines are binding with each other, what is happening? If those two pyrimidines are binding with each other, what is happening? In case of if adenine is binding with guanine, it is a lot more wider structure because they have 4 rings. If cytosine will bind to thymine, then it is very narrow. But they have found that DNA have a continuous straight structure. The width is same all the time. Then what is the composition pattern there in the DNA? Only the possible cause is that one purine should bind with one pyrimidine, and that is constant. Okay? Because 2 and 1, they are forming the 3 rings in one step. It is not between purines, not between pyrimidines. Later we have found that only adenine is binding with thymine or uracil and guanine is binding with cytosine only. So that composition is okay. That is one purine with one pyrimidine all the time. Now, there are 2 terms. One is called nucleoside, one is called nucleotide. When it is nucleotide, that means the phosphate group is there. When it is nucleoside, that means the phosphate group is not there. So this is actually nucleoside, this part. When the phosphate is there, then it is nucleotide. And based on the number of phosphates, there are 3 types of nucleotides. One is monophosphate, diphosphate, or triphosphate. These are 3 things abundant in cytosol. Okay. So these are the primary structures. So we have the bases, we have the sugar base, sugar, base, and phosphate group. But what is the structure actually of the DNA? DNA is double helical. That means there are two anti-parallel strands. One is from this way, another is from that way. And the backbone of this anti-parallel strand is for phosphate groups. They are forming the phosphodiester bonds. And between the steps of the ladder, each and every step of the ladder is made up of nitrogenous bases, that is the nucleotide bases, and these bases are joined by hydrogen bond. And this is the structure of that. So anti-parallel. Anti-parallel means they have two opposite structure. Here is the prime 5' position. Sorry, I told that 3' is OH and 5' is phosphate. So in the 5', there is phosphate group here in this side. In the anti-parallel strand, it is in the opposite side. Okay. So if one strand is 3 to 5, then other strand is 5 to 3. And that is very, very important for DNA replication. Okay. This part, these are the strands, they are made up of phosphodiester bonds. Only the phosphate groups are attached to the next OH with the bond, phosphodiester bond. Each and every step with the nucleotide bases. And purine C is bonding with G, A is bonding with T. Between this bonding, there are just hydrogen bonds, and these are easy to break. In case of RNA, RNA is single strand. Generally, in some cases, the double-stranded RNA is also present. But generally, RNA is single-stranded. But RNA can make their structure, three-dimensional structure. In case of tRNA, transferred RNA, we can find that their structure is three-dimensional. They can form the bond between themselves. Only one strand, but they can form the bond between themselves and can form the three-dimensional structures. Okay. In case of some bacteria, we have double-stranded RNA also. Now, based on this structure, that is, the structure has some kind of different things here. Based on the structures, we have three types of DNA. One is called aDNA, one is called bDNA, and one is called zDNA. Generally, the most common DNA, what we have, or what Watson and Crick described, that is called the bDNA. Okay? bDNA is something like that. There are two anti-parallel strands are there. In one side, this is the 5' end, so that means it is 3' to 5' in this way, and this one is 3' end OH group, so 3' to 5' in this way. Okay. Each and every ladder, each and every base pair is 0.34 nanometer width. Each and every base pair, the difference. These are called two groups. One is called the major group, that is the bigger one. One is called the minor groups. The major group is generally, this is the one turn for the major group, and each and every one turn, there are 10 bases of DNA. If the difference between two bases is 0.34 nanometer, that means in one turn, they are 3.4 nanometer in length. They describe the whole thing clearly in this way. And the width of the DNA, that is from this part to this part, and that is 2 nanometer always. And that tells us that purine is joining with pyrimidine, that is AT and CG base transfers. These are three forms of DNA. The two are right-handed turns, that is the aDNA and bDNA, right-handed turns. And one is the left-handed turns like this. This is called the zDNA. Okay. So these two are right-handed turns, but their difference is that their major group is much bigger than this one. This is called aDNA and this is called the bDNA. Their major group is much wider than the aDNA, the normal bDNA. Generally, aDNA is found in some kind of bacteria. But cDNA and vDNA both are available in human cells. In case of zDNA, which is in opposite direction, but having the similar structure like vDNA, just in opposite directions, they might cause some kind of that is mental disorders. Watson Crick proposed another model which is called central dogma. What is central dogma? Central dogma means DNA can transform into another DNA. This is the first thing. DNA can produce another DNA. This is called DNA replication. Then DNA can express through proteins. The first part, DNA can produce RNA, that is called transcription. RNA can produce protein, that is called translation. This model was proposed by Francis and Crick, and this is called central dogma model. Okay. After that, during 1950s and 60s, we only know that the DNA can replicate into DNA, DNA can produce mRNA, mRNA can produce protein, up to this. Now we know that DNA can produce RNA, that is the okay, but sometimes RNA can replicate by themselves in some other bacteria and some other areas. RNA can replicate by themselves. Not only that, in some bacteria, which are the RNA bacteria, they can produce the DNA. So there is a chance of producing DNA from RNA. That is called reverse transcription. This is called reverse transcription. Using this reverse transcription, right now we can produce the complementary DNA or cDNA. Because our target is right now, the target is proteomics and transcriptomics, that means RNA. So we are taking out the RNA from the human sample or human blood. From that RNA, we can use the reverse transcriptase enzyme and can produce the DNA. Because DNA is stable, RNA is not stable. RNA is very, very unstable. Even when we are working with the RNA samples, we have to use 2 gloves because from our skin cells, if RNAs go to the sample, they can destroy the whole RNA. So we have to use 2 gloves, full lab coats and everything. Okay, so that's why we need to prepare the cDNA or complementary DNA from the RNA, and that is using reverse transcriptase enzyme. Okay, up to this today. Then we will go a little bit about special natures, then chromosome structure, and then DNA replication. This model, you need to know it very clearly. Just go to the DNA structure, because in case of DNA replication, it is very, very difficult. That is one of the tough subject.